Chapter 12 is all about antimicrobial treatment. So now we're talking about ways to control growth of microbes in the body. So a couple of vocabulary words to start. We have descriptors of different drugs that are used for controlling growth of microbes inside of a person. If a drug is described as being an antimicrobial, then it is a substance that interferes with growth of microbes, and that substance can be synthetically, semi-synthetically, or naturally produced. So the synthetically produced drugs are made in a laboratory. The semi-synthetically produced drugs are derived from a natural compound that has been altered in a laboratory, and the naturally produced are compounds that are made in nature and have been isolated from nature that are found to have antimicrobial properties. Antibiotic, which is the phrase we normally use for drugs and we kind of misuse, are substances that control growth of microbes in the body or in a host. But all antibiotics technically are naturally produced by another microbe. So there is no synthetic or semi-synthetic drugs that are called antibiotics. They would be referred to as antimicrobial. So antimicrobial references synthetics, semi-synthetic drugs, and the naturally produced antibiotics. When designing an antimicrobial drug or selecting one, there are several factors to consider. So this is a list from your book that kind of encompasses all things to be considered. And then we also have a few tests that can be done in a laboratory to get an idea of what the type of microbes affected by the drug are, what concentration we should use, and then can determine some things about toxicity. So we'll start with the first of the tests, which is the Kirby-Bauer disc diffusion test. So the point of this test is to determine if an antimicrobial can kill a specific bacteria. So this is figuring out what the spectrum of activity for the drug is. So how many different groups of microbes can the drug kill? So this is what a Kirby-Bauer disc diffusion test looks like. So in this test we have a petri dish which contains a layer of agar containing all essential nutrients for the growth of a microbe. We have a microbe culture that is spread over the surface of the agar on the plate. And we have paper discs which are soaked in different antibiotics. So to set up a Kirby-Bauer disc diffusion test, you take the agar plate, you would take a culture of bacteria or some other microbe, you would spread that microbe over the surface of the agar in a very even fashion so that you would ideally get growth on all areas of the agar plate. Then you overlay the area that you've just spread with the microbe culture with the paper discs. So you put the paper discs over top of where you've just spread the microbe, and each of the paper discs, having been soaked in antibiotic, will be left overnight to allow the microbe to grow and also to allow the antibiotic from the paper disc to seep out into the agar in a radial fashion. So it will create a zone around the disc that contains antibiotic that gets into the agar. And as you move further away from the disc, the concentration of antibiotic will decrease. So in this Kirby-Bauer experiment, we can see areas where the bacteria is growing. They look slightly whitish. And then around some of the discs, we see areas where the bacteria is unable to grow. So these areas are called zones of inhibition. So let's look at the zone of inhibition around the disc labeled TE30. So this is a disc that has been soaked in the antimicrobial tetracycline. 30 is referencing the concentration of the antimicrobial. So overnight, the tetracycline antimicrobial drug has seeped out into the agar in this circular way, and everywhere that the tetracycline is in the agar in a high enough concentration, the bacteria is unable to grow. So you can see this whole circular area around the disc with no growth. So a conclusion from this experiment would be that 5 of these antimicrobials, so we have 2 on the bottom left and then we have the 3 in the upper right corner, all have a zone of clearing or zone of inhibition around them. So each of these antimicrobials appears to have some activity against this specific bacteria. So in order to quantify the Kirby-Bauer disk, after it's incubated for 24 hours, you would then take it out of the incubator, which would be set at a temperature at which the bacteria ideally grows. And you would look around all of the disks and try to see if there is a zone of clearing. So in this drawing of a Kirby-Bauer plate, the zone of clearing is drawn as the yellow circle. So we see a really big zone of clearing around enrofloxacin, and then there are varying sizes of zones of clearing around all of the other antimicrobial discs. So you would then take a ruler and you would measure the diameter of the zone of clearing around each of the discs. So with each of these antimicrobials, there is a zone of clearing that is considered the limit. So if the zone of clearing is bigger than the limit, then the antimicrobial is going to be effective against against the microbe, and the microbe will be susceptible to that antimicrobial. If the zone of clearing is non-existent or is below the limit, then that microbe is resistant to that antimicrobial drug. So we're looking for the bigger zones of clearing indicate that the microbe is susceptible to these antimicrobial drugs, and the smaller zones of clearing indicate that the microbe is resistant to these antimicrobial drugs. So in each of these antimicrobials, we see, in addition to the name and the concentration, so we can look at gentamicin, 10 grams. If the zone of clearing is smaller than 17 millimeters in diameter, then we would say that the microbe is resistant to gentamicin. But if the zone of clearing is greater than 21 millimeters in diameter, we would say the microbe is susceptible to the antimicrobial. So if the zone of clearing is greater than 21 millimeters for a particular microbe, gentamicin would be a good candidate for treating an infection with that microbe. Now, there is a zone between 17 and 21 for gentamicin, which is just called intermediate or inconclusive. So if you got this result somewhere between resistant and susceptible, you would do further experiments to determine if the microbe is sensitive or resistant. You can also see Kirby-Bauer disk diffusion test results written out in a table like this one. So in this table, we have 6 different antimicrobial drugs listed on the left. We have their zone sizes required for the microbe to be susceptible or for the microbe to be considered resistant. So we have each of these antimicrobial drugs is paired with a bacteria. So in this case, it's Staphylococcus aureus. And so we do a Kirby-Bauer experiment. We incubate overnight or for 24 hours. We then look the next day and we measure the diameters of the zone of clearing around each of the antibiotic-soaked discs. And we see the measurements listed in the third column. So let's look at neomycin. The zone of clearing around the neomycin disc was 12 millimeters in diameter. So then if we look at the zone sizes, we see 12 millimeters is right at the limit of resistance. So Staphylococcus aureus is resistant to neomycin. We can look at the last line, vancomycin. So the zone of clearing around the vancomycin disc on the Staph aureus plate was 15 millimeters in diameter. So if we take that number and look at the zone size requirements, we see that 15 is greater than 12, so we're in the range of susceptibility. So Staphylococcus aureus is susceptible to vancomycin, meaning we could treat a Staph aureus infection with vancomycin, but we would not select neomycin because Staph is resistant to this antimicrobial. Okay, the second test that we do in the laboratory to kind of characterize a antimicrobial with a particular microbe is the minimum inhibitory concentration test, or MIC test. So the point of this test is to get an idea of what concentration of the antimicrobial is required to prevent growth of the bacteria. Okay, so a minimum inhibitory concentration test is shown in this slide. So we have several test tubes which are filled with a liquid media. So this is a liquid that has all essential nutrients and requirements for microbial growth. The media is going to also contain varying amounts of a particular antimicrobial drug and will contain a particular microbe. So in the negative control tube, we have the media, we have the microbe growing, and we have no concentration of the drug. In the tubes subsequently, we have increasing concentration of the drug. So starting at 0.2 micrograms per milliliter, we go all the way up to 12.8 micrograms per milliliter. So the growth of the bacteria is indicated in yellow. So any tube where the bacteria is able to grow is colored yellow, and tubes where the bacteria is not able to grow is colored white. So in this particular experiment with this antimicrobial drug bacteria combination, the minimum inhibitory concentration, or the first concentration where bacteria growth is prevented, is 6.4 micrograms per milliliter. So above that concentration, we do not see growth of this bacteria in the presence of the drug. Below that concentration, we do see growth. So we would establish from this experiment that the MIC for this drug bacteria combination is 6.4 micrograms per milliliter. MIC tests can also be done on 96 well plates. So each of these circular wells is going to contain a few drops of liquid. So this is a way to do a lot of testing of many different antimicrobials with different microbe combinations with a small amount of liquid, and these types of plates can be read by machines. So in this MIC experiment, the color change is from pink, indicating growth of the microbe, changing to blue, indicating no growth of the microbe. In each row, we have a different antimicrobial drug. So AMB is in the second row, FLUC is the drug in the third row, and so on. We also have listed the starting concentration of the drug, and then in subsequent wells, that concentration of drug will double. So in each of the wells, in addition to adding the drug and the media, we add the microbe. So in the first antimicrobial, AMB, we see growth of the microbe in the first column, the second column, the third column, and the fourth column. But when we get to the fifth column, we have an X because the microbe is no longer able to grow at this high concentration of AMB, antimicrobial drug. So we would establish the MIC for AMB with this microbe as the concentration of AMB in the fifth column of the second row. The positive control row at the top would just be one well containing the microbe and the media to ensure that the microbe is growing as you would expect. So you can see at different concentrations, all of these antimicrobials are able to prevent growth of the microbe. FLUC antimicrobial requires a very, very high concentration in order to stop growth of this microbe, but it is eventually able to do that. Okay, the third test is the therapeutic index, which is used to determine the toxicity of a drug. So toxicity is a measure of the side effects of a drug. If a drug is very toxic, then it's going to cause a lot of off-target effects. So it may kill the microbe that's infecting the host, but it may also cause damage to the host cells and tissues, which will make the host sick. Ideally, we want an antimicrobial drug that has very low toxicity. The therapeutic index is a ratio. So it is a ratio of the dose of the drug that can be tolerated divided by the effective dose of the drug. So ideally, the dose of the drug that can be tolerated would be very high, in that a person could take a lot of this drug without experiencing any negative side effects. But the effective dose of the drug ideally would be very, very low. So the person has to take a very small amount of the drug in order to deal with the infection. So the therapeutic index, the numerator should be very high, the denominator should be very low. So we're looking for a very high number with therapeutic index. If we have a number that's equal to 1 for therapeutic index of a drug, we see that the tolerated and effective doses of the drug are basically the same. So you will get some side effects probably from taking the drug. If the therapeutic index is less than 1, then the dose of the drug that can be tolerated is smaller than the effective dose, meaning you have to take more of the drug to deal with the infection than the human can tolerate. The only example of drugs with a therapeutic index of less than 1 that are in use are chemotherapy drugs for treating cancer, because we don't have a better way to deal with cancer. So the drug kills the cancer, but it also damages the host and makes the host very sick. So a higher therapeutic index is ideal, and a high therapeutic index means the drug has very low toxicity. We would also consider the spectrum of activity for antibiotics. So this could be established through the use of Kirby-Bauer plates. So some antimicrobials have a very broad spectrum of activity, which is the case with tetracyclines. So the line for tetracycline indicates that this antimicrobial is effective against gram-negative, gram-positive, chlamydia, and rickettsia groups of microorganisms. A drug with very narrow spectrum of activity would be tobramycin or polymyxin. Both of these are effective only against mycobacteria, maybe slightly against gram-negative bacteria. So normally narrow spectrum drugs are ideal because they can target the microbe infecting the host specifically, and will not target other microbes that may be present in the normal microbiota and will be protective for the host. In some cases, if the infection is very serious and life-threatening and the cause of the infection is not known, a broad-spectrum antimicrobial will be prescribed to try and deal with the infection, and in the meantime, the cause of the infection will try to be identified. So broad spectrums are used in life-threatening situations, but they do often disrupt the normal microbiota of the host, which can make the host sick. So this is what would happen if an antimicrobial is taken that does kill the normal microbiota. In some cases, when the normal microbiota is killed or taken out of balance, this can cause other microbiota that are present in the host to overgrow, which will make the host sick, causing a super infection. We are only discussing in this chapter antibacterial drugs. There are sections on antivirals, antihelminthics, and other groups of microbes being targeted through drug treatment. I just want to point out that viral infections are more difficult to treat than bacterial infections because viruses are obligate intracellular parasites. So shown in this image, we have a virus that binds to a receptor on the surface of a human cell. That virus then gets inside the human cell and uses the human cell to replicate its parts and genome so that viruses can escape from the host cell in step 4. Targeting a virus is very challenging because viruses live so much of their life cycle inside a human cell, so it's very difficult to target a virus without damaging a human cell. The way that antibacterial drugs work is through a number of different cellular targets. So different antibacterial drugs will target different aspects of the bacteria cell. So we're grouping these into 5 categories, and we'll go through each one of them briefly now. So the first group of antimicrobial drugs are those that target the cell wall, and these are very specific for targeting bacterial cells because human cells don't have a cell wall. So we have no off-target effects. The beta-lactams are the group of antibiotics that are cell wall targeting in the bacteria. So shown here are 4 different molecular structures of antimicrobial beta-lactam drugs. You'll notice that each of the molecules shares the same 4-ring structure, which is highlighted in orange. This is called the beta-lactam ring. And this part of the structure is really important because the beta-lactam ring can wedge in between peptidoglycan, which is the molecule that makes up the bacterial cell wall, and will prevent the bacteria from being able to build cell wall. So beta-lactam antibiotics are most effective when the bacteria are in the log growth phase, when they're doing a lot of building of cell wall and a lot of dividing. In the presence of beta-lactam antibiotics, the bacteria is unable to build cell wall, and so the bacteria can't replicate and will eventually die off. The second group of antimicrobials are those that affect protein synthesis in the bacteria cell. So I'll remind you that the bacterial ribosome is a 70S ribosome, where the ribosome in humans is 80S. So a lot of these protein synthesis inhibitors will selectively target the 70S ribosome. You do not need to know the specific ways that these protein synthesis inhibitors bind to the ribosome or what they specifically prevent. Just like you to know that these antimicrobials will all affect the ribosome and its ability to build protein in some way. I will also remind you that protein synthesis inhibitors, if taken for a very long time or at a very high dose, can affect the eukaryotic ribosome that's present in mitochondria because it is also the 70S size. Folic acid synthesis inhibitors are targeting a essential metabolite in bacteria cells, so the sulfonamides or sulfa drugs are able to prevent bacteria from being able to synthesize folic acid. And this is a big deal for bacteria because folic acid is a precursor for making DNA and RNA. So when sulfa drugs prevent bacteria from making folic acid, they essentially prevent bacteria from building DNA or RNA, which is essential for the bacteria to function. So the bacteria will die without the ability to build these nucleic acids. So the reaction that we're looking at is PABA being converted to dihydrofolic acid, which is then converted to THF, which is then converted to purine and pyrimidine nucleotides. So this is the A, C, G, T, and U. And then those are built into DNA or RNA. So we're focused on the enzyme that catalyzes the reaction of PABA to dihydrofolic acid. And so that enzyme is shown in the lower left of this slide, and it is shown with PABA bound in the active site. So when PABA binds in the active site of this enzyme, the enzyme catalyzes its conversion to dihydrofolic acid, which it will then leave the active site so another PABA molecule can come in. But with the sulfa drugs, which are now shown at the top of this slide, so we have sulfanilamide, sulfamethoxazole, and sulfisoxazole, which are 3 types of sulfa drugs, compared to the structure of PABA on the left. So you'll see they all contain a H2N bound to a 6-sided benzene ring. So when PABA binds to the active site, it's converted to dihydrofolic acid. But in part C, we see the enzyme with a sulfa drug bound rather than PABA binding. So the sulfa drugs are a competitive inhibitor for PABA binding to its enzyme. And when the sulfa drugs are bound, PABA can't bind to the enzyme, so folic acid is not produced and the bacteria is unable to make DNA or RNA. So because of the similarity of structure with sulfa drugs and PABA, the sulfa drugs can competitively inhibit PABA from binding to its enzyme and being converted to folic acid. And this is a good target for killing bacteria specifically because bacteria synthesize folic acid where humans just get folic acid from our food. So we don't have this enzyme that converts PABA to folic acid. So we will not be affected by sulfa drugs that competitively inhibit PABA from binding. Okay, the fourth group, inhibition of DNA and RNA synthesis. So these antimicrobials are the fluoroquinolones and the quinolones, and they block an enzyme called DNA gyrase that unwinds the double helix so that the replication fork can be formed. The bacterial form of DNA gyrase is slightly different than the human form of DNA gyrase, so these drugs do not have an effect on human cell ability to synthesize DNA or RNA. And then the last group are cell membrane inhibitors. So the example of this we'll give is polymyxin B antimicrobial. So this is a drug that targets LPS in the gram-negative cell membrane. The figure shown on this slide contains more detail than you need to know, but I've added it because it shows polymyxin, which is drawn as a little pink molecule, is able to wedge into the phospholipid membrane layer of the bacteria. And so that's how it disrupts the cell membrane of the bacteria. And because it targets LPS, which is a molecule in the cell membrane of bacteria but not human cells, it is specific for bacteria. Okay, so now let's discuss what bacteria do to become resistant to antimicrobials. So if a bacteria is susceptible to an antimicrobial, the antimicrobial will kill the microbe. But if a bacteria is resistant to an antimicrobial, the drug will have no effect on the bacteria. So if we have a population of bacteria growing on a petri dish, we have the drug-resistant ones shown in red and the drug sensitive or not drug resistant bacteria shown in white. We treat them with antibiotic. What will happen is all of the drug sensitive will be killed off and the drug resistant will now have the entire petri dish and all the nutrients to increase in number and make a whole population of now resistant microbes. So this is what can happen in our body. We can have some microbes are resistant. We take antibiotics, kill off the sensitive ones, and then the resistant ones can take over. So bacteria become resistant to antimicrobials either through acquiring new genetic information, which results in one of these 5 things happening. So one example is new enzymes are made by the microbe that make it resistant to the antimicrobial. So an example of one of these enzymes is penicillinase, which is an enzyme that some bacteria can make that will open up the beta-lactam ring of a beta-lactam antimicrobial so that the antimicrobial drug is now inactive and the microbe can survive in its presence. We have some microbes will reduce the permeability or uptake of the drug by altering the receptor on the surface of the microbes that the drug will bind to. Some antimicrobials can make drug pumps through acquiring new genes, and these will actively remove the antimicrobial from the interior of the microbe. Some can alter the binding site, so the one shown here is the bacterial ribosome, where the drug binds to the lower left part of the large subunit of the ribosome, but the ribosome is altered at that spot so the drug can no longer bind. Or the last example of how bacteria could become resistant to an antimicrobial is if they acquire genes for making a product in a new way. So if the drug is inhibiting the reaction of B being converted to C, which is eventually turned into product, the microbe could come up with an alternative pathway to make the product, so avoid the reaction that is inhibited by the drug. We also have some microbes which are just naturally resistant to antimicrobials. So an example of this is gram-negative bacteria, because they have a double plasma membrane. They have the cell wall and they have the periplasm, which is the space in between all these layers. Some antibiotics can't get into the interior of a gram-negative. So typically gram-negative bacteria are much more difficult to kill than gram-positive. So bacteria become resistant to antibiotics either because they naturally are, as we just said in the last slide, or because they spontaneously have DNA mutations, which would be an error being made when the DNA is copied. And that error would result in some new ability of the bacteria to avoid the action of the drug. Or the bacteria can acquire new genetic information, and this is mostly through horizontal gene transfer. So ways to combat antimicrobial resistance are to use drugs properly, to try to develop more synthetic antimicrobials that are new and that their bacteria are not resistant to yet. Trying to discover new antimicrobials, which is not in the interest of drug companies because antimicrobials are not very profitable because people take them for a short amount of time and they don't take them chronically like drugs for diabetes. There are some studies looking into using lytic bacteriophage to kill bacterial infections that don't respond to any other microbial drugs. And then there's a lot of efforts with public health campaigns like this one from the CDC to try to educate people and limit their use of antimicrobials. And that concludes Chapter 12.